Bismillahirrahmanirrahim. We are doing the 9700 biology paper 1 2. Now this is an MCQ paper and we're going to try and understand it and solve it. So this is paper 1 2 March 2016. Now let's proceed with this paper that we have. Now looking at the first question, it says a student has drawn a cell structure as seen using a light microscope. The magnification of the drawing is x600. The length of the structure on the drawing is 6 millimeter. What is the actual length of the cell structure? Now, if you know it's 6 millimeter, now 6 millimeter is 6,000 micrometer. Now, you know all the answers as you look at it were all in micrometers. So you needed to figure this out. Which one is it? So 6,000, 6 millimeter, this was given in the question, 6,000 micrometer divided by 600 is equal to 10. And that, of course, makes the answer C, which was the correct answer, and this is how we were going to figure this out. Then let's come on to question 2. Now, as you look at question 2, it says the electron micrograph shows part of a eukaryotic cell. And you know eukaryotes are what? Eukaryotes are animal cells, plant cells, and fungus, and protozoa. So all the Protoctista are also, now that if you're taking the composite, you've done A2 as well, and you know what it is. So animal cells, plant cells, fungus, and Protoctista are all eukaryotes. So which of the labeled organelles is a site of protein synthesis? Now, a site of protein synthesis, if you look at A, what is A? It looks like a lysosome. C is a centriole, and D looks like the Golgi apparatus. So if that is the Golgi, and then you say, oh my God, what is A? A is lysosome. Now, what is possible that could be protein synthesis? You see, you have to have protein synthesis can only be where there are ribosomes. Now, where there are ribosomes? There are ribosomes in the cytoplasm. So we have the free ribosomes in the cytoplasm. Then we have the fixed ribosomes on the RER. But then this was not anywhere here. The fixed ribosomes are on the RER. So, but here then we had mitochondria. And mitochondria has 70S ribosomes. So that is where the protein synthesis was going to take place. And it said it's a eukaryotic. And the centriole tells you that this is an animal cell. It's not a plant cell where you could have had chloroplast. And of course, you remember in chloroplast also we have ribosomes. And that is also a place where protein synthesis can take place. So question 2 was B. Then we come on to question 3. Now question 3, inside a cell, a damaged mitochondrion can be surrounded and enclosed by a membrane to form a vesicle. What happens after the fusion of a lysosome with the vesicle? So this is the mitochondrion, and it said a damaged mitochondrion can be surrounded by and enclosed in a membrane. So this is the damaged mitochondrion, and then it said what happens after the fusion of a lysosome. So this lysosome attaches. So the lysosome attaches to this vesicle. So this is the lysosome. So ATP production by the mitochondrion increases. Enzymes from the lysosomes repair the mitochondrion. Hydrolytic enzymes catalyze the breakdown of the mitochondrion. The mitochondrion is released from the cell by exocytosis. So hydrolytic enzymes break down unwanted organelles. So we all know that what is the role of lysosomes, and lysosomes contain hydrolytic enzymes. So that was the answer to question 3. Question 4, which units are the most appropriate to record the diameter of a lymphocyte and red blood cell? Lymphocyte, red blood cells, and you know, of course, it's not in millimeters, it's in micrometers. You must be knowing these, the sizes of the different cells, and you need to look that up and make your notes and revise that. Then coming to question 5. Question 5 was which structures will be present in a cell that causes cholera? Now, you know, cholera is an infectious disease and it is caused by Vibrio cholera. And Vibrio cholera is what? It's a bacteria, and a bacteria is a prokaryote. So which structures will be present? Yes, all prokaryotes will have a circular DNA, and it's a naked DNA. It doesn't have any histone proteins, and it has 70S ribosomes. So 1, 2, and 3, and the answer was A. Then coming on to question 6, what may take place during a hydrolysis reaction? A molecule of water is produced, a glycosidic bond is broken. What is hydrolysis? You have to revise hydrolysis and condensation. Hydrolysis is when you add water, which is given here in this diagram. Hydrolysis is large molecules are broken down apart to produce smaller molecules. Energy is released. Water is used to split the bonds. So the water ends up into H and OH, and this bond is broken. So a molecule of water is produced, a glycosidic bond is broken, a sucrose molecule is split into fructose and glucose. So a molecule of water is not produced. This was the mistake in it. A molecule of water is added. So you see this water molecule being added to it because the water molecule splits into H positive and an OH negative. So the water splits into hydrogen and hydroxyl group. So this is how you had to understand it. A molecule of water is not produced. A molecule of water is produced in condensation. Now I could change this question and I could say what may take place during a condensation reaction. And in a condensation reaction, it will be only one, because a molecule of water will be produced. So this is how we can change the MCQs and make it a new MCQ by just changing one word there. And of course, then the answer would only be one. Of course, there is no answer one only here in the possible answers. Then we come on to question 7. Question 7 is the table shows some information about the table shows some information about 4 carbohydrate polymers. So polymer 1, 2, 3, 4. Alpha 1, 4 glycosidic bonds, alpha 1, 6 glycosidic bonds, shape of molecule, and which two polymers form starch. Now, starch is made up of what? Amylose and amylopectin. Now, the diagram here that I've given you. So, amylose and amylopectin. And amylose has 1, 4, and amylopectin has 1, 4, and 1, 6. So, this is how you were going to figure out it was 1 and 4. So please just revise amylose, amylopectin, and what is the part of it, and then of course you'll be able to figure this out. The branched is only amylopectin, and the branching is due to the 1,6 glycosidic bond, and then of course it's all 1,4 after that. So it's only here that you can see this one here, 1,6, and then of course they're all 1,4. And coming to question 8, which molecule in the key is sucrose? And I've given you this information that sucrose is a disaccharide made of glucose and fructose. So first it says it's a reducing sugar, then contains a pentose sugar, then contains hexose, and then yes, no, and all that. Now, so which one it could be? Pentose, A, yes, no, yes, no. So of course it had to be contains hexose because both glucose and fructose are hexose sugars. So it had to be the answer was C, which was contains hexose, and because why? Reason is sucrose is a disaccharide made of glucose and fructose. Okay, then we have which statements about triglycerides and phospholipids are correct? Triglycerides and phospholipids both have a hydrophobic region. Triglycerides are non-polar molecules and phospholipids are polar. Fatty acids in a triglyceride may be saturated or unsaturated, but in a phospholipid they are always saturated. Now, as I have underlined it, it's a lot of BS. Of course, BS means beautiful story. So, the answer was 1 and 2, and the 3 was a big BS because fatty acids in a triglyceride may, but in a phospholipid they're always saturated. No, in a phospholipid bilayer we have lots of sometimes saturated fatty acids, sometimes unsaturated fatty acids. So that's why I've given the BS, which is the beautiful story, which is why 3 was total rubbish and this was wrong. So the answer to this was 1 and 2. Then we come on to the question 10. Now, if we look at question 10, when proteins are mixed with some organic solvents, hydrophobic interactions and hydrogen bondings are changed in the protein molecule. Which level of protein structure would be affected? So we have primary, secondary, tertiary, quaternary, but here we only had 3 levels which were discussed: secondary, tertiary, and quaternary. So hydrophobic interactions and hydrogen bond. Now, if you know, hydrogen bonds are present in the secondary as well as the tertiary, and of course they have also, so the answer to this was also the quaternary would be affected, and I've given you a whole list of this, which of course I will discuss with you at the end as well, which I will talk about this diagram here, which I have given you here. So primary structure, and this I'll go at the end, I'll discuss it again. Then coming to question 11, the rate of enzyme catalyzed reactions in humans is regulated. What may be involved in such regulation? So the rate of enzyme catalyzed. So now, of course, the question is rate of enzyme catalyzed reactions in human cells is regulated. Number one, a change in the enzyme concentration. Now you see I've drawn this diagram and I've shown you that there are 10 of these and there are 50 of these as a substrate. So a change, if there instead of 10 there are 100, what will happen? Then a change in the substrate instead of 50, if there are 500 or 5,000, then inhibition by the final product of the reaction, and that is called end product inhibition. So it has to be all three would be affected, because if you increase the number of active sites, the rate of the reaction will go faster than if you increase the number of substrate. Naturally, more product will be formed per unit time, and more enzyme means even further. If you increase the enzyme's active site, more product. But if you increase the substrate, after some time the substrate will finish and the rate will stop. But if the substrate is in excess, the enzyme reactions are going to keep on taking place. But then in some situations, there is end product inhibition. After a series of reactions, sometimes the end product is going to inhibit the first enzyme, and then that's going to stop the reaction from occurring. And I've given the example of this in my video on the chapter on enzymes. Looking at question 12, it says the graph shows the effects of temperature and pH on enzyme activity. Now, as you can see, the rate of reaction, and then we have P, and then we have Q. So we've got this here, we've got this here, and then we've got R here, and then we've got S here. Now, what you don't see is that temperature, the highest energy will be somewhere here, because the temperature is increasing. So I've given 10, 20, 30, 40, 50, 60. So the kinetic energy would be the highest here. And pH, let's just write anything on it. 1, 2, 3, 4, 5, 6, 7, anything. Do that all the time. Whenever you're doing the graph, write on the graph, give some figures on the graph. Similarly, you could give some figures here as well. Rate of reaction 10, 20, 30, 40, 50, something like this here as well. And then, of course, it said which statement is correct explanation of the enzyme activity. At P, hydrogen bonds are formed between enzyme and substrate. At Q, the kinetic energy of the enzyme and substrate is highest. That's rubbish. The highest is at here at this point. At R, disulfide enzymes in the bonds break, and the enzyme begin to break. I mean, why? Disulfide is the tough bond. It doesn't break very easily. At S, the enzyme is completely denatured. No, it's not completely denatured. At S, it's still continuing its effect till the last bit here. So we have to understand is what they're asking, and you have to read the graph very carefully, and you have to interpret the graph very carefully. Then it says which statement correctly describes the action of competitive enzyme inhibitors. Now we are on question 13. They bind permanently to the active site. Competitive means that the shape is the same as the substrate. So you can see I've given you this. So they change the shape of the active site. No, they limit the formation of enzyme substrate complexes. Naturally, you have 10 of these, 10 of these, and 10 of these. So I mean, these can also fit, and of course no product is formed. But if this fits, then the product is formed. So please read the question very carefully and understand it, and make these sort of diagrams to explain it to yourself, and you will figure out the answers very easily. Coming to question 14. Now if you look at question 14, which row correctly links molecules in the cell surface membrane with their roles? So barrier to polar molecules, stabilizes membrane, receptor. Now, of course, we have to understand is that 1, 2, 3, 4, and how they've linked it. So 1 could be what? Which is common to the barrier to polar molecule. The barrier to polar molecules is only the phospholipid layer. You see, you've got to understand is that this is the phosphate head. These are the fatty acid tails. And this is the part which is not going to allow polar molecules to pass. So barrier to polar molecules is the fatty acid chains. And stabilize the membranes is, yes, many like cholesterol stabilizes the membrane. Receptors are glycolipids and glycoproteins. So of course the answer is D, and this was something you had to nearly revise the cell membrane chapter. Then it says antimycin is a chemical Antimycin is a chemical. Now we are on question 15. Antimycin is a chemical that inhibits the function of mitochondria. Which method of transport across the cell membrane would be inhibited by antimycin? Active transport is an active process. Facilitated diffusion is passive. Endocytosis is active. You see, you have to realize mitochondria produces ATP but releases energy. So these are the words that we use biologically, which is correct English. So mitochondria produce ATP, but if we have to use any other word, we say releases energy. So which method of transport will be affected? So it's only the active processes, active transport and endocytosis. So that is why the answer was 1 and 3 only. Then we come on to question 16. Which statements about the cell cycle are correct? The cell cycle consists of both interphase and mitosis. DNA replication takes place in interphase. A cell can remain in interphase for several months. Well, that's very correct. So all three are correct. So interphase and mitosis, though I would say the cell cycle consists of interphase, mitosis, and cytokinesis, but of course they have talked about cell cycle. So cell cycle is these both are present in the cell cycle. So it's 1, 2, and 3. We come to question 17. The diagram shows cells in different stages of the cell cycle. Which is the last stage before cytokinesis? Now, I always tell you to remember this mnemonic. Interphase, prophase, I have a purple mat. So, and then of course it is cytokinesis. So I don't call that here. This is all part of mitosis. So this is all mitosis. So interphase, prophase, metaphase. Now, but they said the diagram shows cells in different stages of the cell cycle. So which is the last stage before cytokinesis? That has to be what? That has to be before cytokinesis. So prophase, metaphase, anaphase, that has to be telophase. Now in this, you might think this is telophase. No, but these are two separate cells. So in telophase, the cell is one, but there are two nucleuses. So we have to understand what we are trying to be. So before the last stage before cytokinesis. So this is metaphase, anaphase. Of course, this is not written in it, but I have just written these on top of it. So telophase. Then coming on to question 18. How many of the listed structures typically contain genetic material that has telomeres? Bacterial cell has a loop of DNA. Chloroplasts have a loop of DNA. Mitochondrion has a circular DNA. So you got to understand is how many of the structures typically contain that has telomeres. So 1, 2, 3, and 4. 1, 2, 3, and 4. So this is of course the answer was. I think there is a mistake in this. So, I mean, I just read that incorrectly. How many of the listed structures, I mean, there are four listed structures, how many of them contain genetic material that has only one, which is the nucleus only has chromosomes, which have telomeres? The rest, so how many of the listed structures? So, if we say this is 1, this is 2, this is 3, this is 4, so one of these only the nucleus contains. So, this is, of course, it's reading the question. I also misread it initially. Then coming on to question 19. Now, what does the enzyme DNA polymerase synthesize in a cell? A polypeptide using DNA as a template, a strand of DNA using a polypeptide as a template, that's all rubbish. A strand of DNA using DNA as a template. A strand of mRNA using DNA as a template is called transcription. So that is something that we need to understand. Now, I could change this question and I could say, which is the statement which is correct for transcription? And of course, the answer would have been D then. A strand of mRNA using DNA template is transcription. So this is how from one MCQ, I make a number of MCQs and see how well you've understood it. Then it says in question 20, 21.2% of the bases in a molecule of DNA are cytosine. So what percentage would be adenine? So if we have 21.2% cytosine, then that means 21.2% is guanine. So 42.4, so 100 minus is 57.6, and adenine plus thymine is 57.6. So what percentage would be adenine? Is the half of 47.6? And that is 28.8%. So that's a bit of math that you have to do. So 21.2 are cytosine. So the same would be guanine. So if this is 21.2, then this is also 21.2. Then we have A and T. So this 21.2 plus 21.2 is 42.4. And so 100 minus 42.4, whatever is left, and then you half that. Then question 21. One gene provides a code for the production of which type of molecule? Actually, everybody knows that one gene provides a code for the production of one type 1 polypeptide. So basic unit of heredity, a gene is a segment of DNA chromosome that describes how to make a certain protein. Genes are located at a specific locus on the chromosome, and genome is the whole of the genetic information of an organism. Then question 22, the table shows the mode of action of two antibacterial drugs that can affect the synthesis of proteins. So we have antibacterial drug rifampicin, antibacterial drug streptomycin. Mode of action binds to RNA polymerase and causes errors in translation. So if there are going to be errors in translation, then we're going to have faulty proteins. So if bacteria are treated with both drugs, what will be the immediate effects? Transcription will stop, but faulty proteins may continue to be synthesized. If translation has started, proteins may be faulty. Translation will be inhibited. So you have to understand is both the drugs, and what do both the drugs do? One binds to RNA polymerase and one causes errors in translation. So the answer to this was 1 and 2. Why? Because it is going to result in faulty proteins. So this was the answers to 22. Then we have 23. Now 23 is a question on transport in plants. The diagram shows transverse section of parts of a plant. In the transverse sections, which tissue transports sucrose? Of course, we know it is the phloem, so you have to identify the phloem. And the answer to that was C. Then question 24. Which statement describes movement through a plant in the apoplast pathway? Now, apoplast pathway is through the cell wall. I always say along the cell wall, apoplast, or through the cell wall, and symplast is through the cytoplasm. And you can see here, this diagram shows you the apoplast pathway. So water moves through the cell walls. When it moves through the cytoplasm, that is the symplast pathway. When it moves through the plasmodesmata, it is diffusion of water. There's no osmosis because there's no cell membrane which it crosses. And when water moves through the vacuoles, that is called the vacuolar pathway. So this is what we had to understand about question 24. In question 25, which changes to the water potential and the volume of liquid in the phloem occurs when sucrose is moved from the phloem sieve tube to an actively dividing root tip? So naturally, when we have to understand is that when the phloem, what were the headings? Water potential in the phloem sieve tube becomes, and volume of the liquid in the phloem sieve tube. So naturally, what it says changes to the water potential and the volume in the phloem occurs when sucrose is moved. So if the sucrose moves out, sucrose is solute, so there are more free water molecules left, so it becomes less negative. And then the water follows, so volume of the liquid also decreases. So the answer was 1. It was A, sorry. Then coming to question 26, it says the diagram shows the results of an experiment using leaves with the same surface area from two different species of plant. Each leaf was left on a balance in daylight in a closed room and its mass recorded at 1 hour intervals. Now, as I always say to you in the graph, label something on the graph. The mass of the leaf was 2, 4, 6, 8. I've just labeled that. And then there's leaf 1. From 8 it becomes sort of 5. And the other one from 8 becomes something like 2. So Which feature of leaf 2 could explain these results? More stomata per unit area of the leaf, fewer trichomes on the leaf, sunken stomata, thinner cuticle. So you see, 2 has lost more water. So what are the factors which make it lose more water? So of course it was 1, 2, and 4. So it has lost more water. Sunken stomata would conserve water. It won't let so much water be lost. Maybe leaf 1 had sunken stomata because it didn't lose so much water. So the same question, I could change it and I could say, what features of leaf 1 could explain these results? And you have to only say 3, because you see leaf 1 lost less water. So I finish this paper here and I'll continue it in the second video. The remaining questions, which are from 27 onwards. And thank you for now.